So I want to tell you a story about some unexpected membrane dynamics that we found in my favorite cells, the killer T cells. So here you can see a killer T cell with the life actin sort of as it interacts with the blue cancer target that it's trying to attack. So if I can get this to move along, there we go. So killer T cells are inherently important cells because they're important in protecting us in the immune system. And of course, they're now forming the sort of revolutionary changes in cancer therapy and immunotherapies. But underpinning their remarkable abilities is the absolutely most fascinating cell biology. And that's how I got hooked. So they use polarized secretion, they're highly dynamic, and as I'll tell you in a bit, they're serial killers. So just to show you the basis of a couple of aspects of the polarized secretion, you can see that they form an immune synapse where the T cell receptors that recognize the target cells cluster upon recognition. And then there are specialized secretory lysosomes that contain additional proteins, in particular perforin that punches holes in the target membrane, allowing granzymes in to trigger apoptosis in the cancer cells. So what I've been trying to do for over I don't know quite how many years now, is to understand the molecular mechanisms that control cytotoxic T cell function and how they achieve such remarkably precise polarized secretion. So just to show you that they are really dynamic cells rather than showing you stills all the time, then I wanted to this movie illustrates how precise the delivery is. So here is a cytotoxic T cell with the actin cortex shown in LifeAct in green, the secretory lysosomes marked by CD63 in red, and then the centrosome, which directs the secretory lysosomes and organizes the microtubules along which they move. And that's marked with a little blue golf ball, really, to show you where it goes. So what you'll see is initially there was an accumulation of actin across the interfaces with the target, then that depleted, and at that point, the centrosome came forward, bringing with it the secretory lysosomes in the most remarkably precise delivery. So we decided to, so the superpower of cytotoxic T cells, as I call it, is the fact that they're serial killers. And this means that they are able to kill one target after another. And here is a short video just showing that this T cell here with the centrosome marked has just killed this target here. And as soon as it's done so, it's able to depart through mechanisms we don't entirely understand and contact another target and polarize its centrosome and deliver more granules and kill it again and again and again. So this increases their effectiveness in the immune system remarkably. So what I want to tell you about today is how we've been trying to map the intracellular dynamics that lead to secretion. And in doing so, we decided to take advantage of this ability to use spinning disk microscopy to do a lot of imaging in 4D, where you can capture the interface between both cells, and you can do so after time. And so we took a model which is where all the T cells have an identical T cell receptor, where you can give it a peptide so it gives an identical level of signaling. And so we can then compare one experiment to the next. And we could look with different combinations of markers and you've expressed within the T cells to identify from the point where the T cell first meets its target, and you can see here a bit of T cell receptor denoted by apex labeling here, just as it begins to contact the target in these lamellipodial projections, all the way through to when the secretory granules are delivered at the synapse. And using this system, what we found remarkable was it only takes 6 minutes for all of this to happen. But during this time, the T cell receptor activates, you get a calcium flux that triggers activation of phospholipase C gamma, and it cleaves PIP2 to generate diacylglycerol. And you get the depletion of PIP2 and actin across the synapse as the centrosome begins to move, delivering the granules so that they can secrete their cargo and destroy the target cell. This is great time, as I always say, because it's just long enough that you can convince your students and postdocs to do multiple experiments and then without them getting too frustrated. But it's also long enough that you can dissect out all of these different points. And my ambition is to dissect this out in as much molecular detail as I possibly can before I die. So I think, so as we began to do this, we noticed that one of the things that rather surprised us was the fact that transcription factor translocation occurs very early. And you think, why do you need new transcription when you've got these beautifully loaded granules, which are going to deliver a massive lethal hit. So we began to ask, we being Ariane Richard, who was in the lab at that point, what is the signature of what we're going to call this early transcriptional burst? So just to remind you, this is what it looks like in the movie, that you suddenly get the influx of this case NFAT, which is what we've looked at, and NF kappa B does exactly the same thing. So Ariane, who's greatly skilled in single cell RNA-seq, in this case used bulk RNA-seq, and she found that actually the early burst of transcription, the dominant RNAs that are expressed are chemokines and cytokines. And these are, for example, these two can drive migration and recruit other T cells to the site where the T cells have become activated. So they're useful in killing. And then, for example, gamma interferon on TNF give additional pathways of killing. But the real question was then, what happens if you stop this early transcriptional burst? How much do you need it? And so that's relatively easy to do because you can inhibit with actinomycin D. And this reduces the, it doesn't ablate, but it reduces the killing. So it says, yes, you need it. But what was really interesting is at the time, we had been doing a lot of killing assays for a screen of different mouse mutants. And the people in the lab were getting sort of repetitive strain injury from all the pipetting. So we tried to find a new method that didn't require quite so many pipetting steps. And we struck upon an instrument called the Incucyte, which measures the killing by imaging. And this turned out to be incredibly helpful because what you do is you take your target cells and you express in them a lentiviral construct, which gives a red signal within the nucleus. So it's a red nuclear label. And then you can make yourself a little artificial pellet of cancer cells by pelleting the cells in your round bottom dish. And then you can add the T cells, which are unlabeled in this case. And what you will notice in the images then over time, we've just shown several of the images here. And what you can see is that over time, normally in the untreated cells, the T cells infiltrate as they destroy the target cells. And the Incucyte calculates the number of targets left. But if you add actinomycin D and inhibit this early transcriptional burst, they can't infiltrate. So this was kind of cool because it told you that the early transcriptional burst is needed for infiltration. And in fact, the lab of Matteo Biro in Australia had shown that you also need it for T cells to recruit other T cells if there are more surrounding. So it's important for killing, particularly when you're looking at solid tumors. But then you have to think, well, how is the early transcriptional burst coordinated with the target recognition? You've got to orchestrate all of these different intracellular arrangements within the T cell so that you get this perfect, precise, and timely delivery of the effector functions of the T cell. So this actually just came out this very last Friday, in the current issue of Science Immunology. So you can look at the movies again and again to your heart content after this. So we began to look about at the nucleus, which we curiously hadn't really done until that point. And I asked Yukaka, we'd always had this idea that maybe the centrosome rolled around the nucleus. And so this is led by Yukaka Asano in the lab. And so she started taking movies using lamin as a marker for the nuclear membrane. And here you can see the centrosome. And here is the target cell labeled with a intracellular dye. So, and what you see, much to our surprise, was that the nucleus polarizes. It goes all the way from the back of the cell to the front of the cell. And I'll just play that again so you can see it. And I want you to compare what's happening with the centrosome. So there's the centrosome, and there's the nucleus. So it's not only that, but the nucleus gets there first. It's way faster than the centrosome, which was a real surprise to us. So you can see it really nicely in these stills from that movie. So you can see here is that this is the projection. But what's really interesting is to look at the Z-slice, where you can see the remarkable indentations that occur in the nuclear membrane and how, as it focuses on the contact site with the T cell where the actin has depleted, that the nucleus really kind of scrunches up. And then eventually the centrosome comes around and gets there too. So you can see this. So we asked, well, how does it occur relative to the T cell receptor? Is that where the T cell receptor is? We might expect it to be there because it's where the actin depletes, but we could show that by using labeled CD3 zeta, which is part of the T cell receptor. And you can see that the CD3 zeta, which is all around the membrane, begins to cluster after about 100 seconds. And you can see it here. Again, you can see things rather clearer sometimes in the Z slices. And so you get the clustering of the T cell receptor at about 100 seconds, and then later you get the nucleus comes, and it focuses at exactly that point. You can see it particularly clearly here. So what does it look like if we zoom in and look by electron microscopy? And this was work from Jane Stinchcombe. And what Jane could see is that actually if we focus on this side first, that you can see if you outline the nuclear membrane, this is from a tomogram, in this particular tomogram, she had labeled where the T cell receptor was by using CD3 zeta with apex in expression in the target, which we always color blue to make it easier to distinguish. And you'll see that the nuclear membrane really comes up and it absolutely contacts this point where the T cell receptor is clustered. Exactly how, we're yet to find out. The other remarkable thing to notice is that actually the nuclear membrane is really invaginated. It's as though it puckers up and somehow is able to focus on this precise spot within the immune synapse, which again is fascinating and yet to be fully understood. So we could then ask, well, okay, this is not what we're expecting to see. It's quite remarkable. I don't think there's another system, although there are other systems where the nucleus moves, I don't think they move quite as dramatically as in these T cells. So we could say, well, where does this nuclear polarization fit into the timeline that we've been making? So where does it occur relative to, say, the calcium flux, the diacylglycerol generation, etc.? And so we would compare all of these events using the different combinations of probes for each of these things. So, for example, with calcium flux, what Yukaku did is she used a trick to just put the GCAMP calcium probe with a nuclear localization signal, so that when the calcium fluxed, that you would see the nucleus as well as the calcium flux in the cells. For us, it doesn't matter whether you put that calcium sensor in the cytoplasm or in the nucleus. It occurs at exactly the same time in the T cells. So if I play this movie, whoops, you will see, I hope, that you suddenly get that. Sorry, misbehaving here. Let's try again. You will see that there's a calcium flux, and the microtubule organizing center, which you can see as the intense pink spot, is still at the back of the cell. So the nucleus starts polarizing after the calcium flux. So again, you can see it down here very clearly. Here's the microtubule organizing center or centrosome, and the calcium flux occurs when the nucleus and the MTOC are both at the back of the cell. And so that tells us that the nuclear polarization is after the calcium flux. And so using this kind of approach, we then were able to compare the timings from the point of first contact. It takes a lot of movies because you have to capture that T cell as it makes its first contact and hope you get the good movie that takes you all the way through. So we could get the timing from multiple movies and show that the calcium flux and T cell receptor clustered very early on. And then it was the nucleus within 2 to 3 minutes that then came all the way and contacted the synapse at that point. And this is way before the centrosome, which is about at 6 minutes. And then one of the reviewers asked us, well, actually, when does the nucleus leave? And one of the interesting things is that what Yukaku saw from the 7 movies where she could capture both events was that the nucleus actually remains at the synapse in all of those movies until the centrosome docks, and then it begins to retract. And so it's about 12 minutes after the first contact. So it comes, it stays quite a while, waits for the centrosome, and then it retracts again. So how does the nucleus move to the synapse? And hopefully Daniel St. Johnston, who I think is on this webinar, will recognize his paper here. We were inspired by his paper, and we thought that maybe it was rather like what he had seen in the oocyte nucleus, where the nucleus polarizes, and it does so by being pushed by microtubules, and you could see nice indentations in the back of the nucleus. And we were struck by these remarkable indentations that we saw in our T cell nucleus, and we said, okay, so maybe it's microtubule pushing. And so here are the growing microtubules, which seem to push indeed into those little dents that we see in the back of the nucleus. So this looked really hopeful. However, when we depolymerized the microtubules with nicotazole, we found that actually the nucleus still polarized, and in fact the actin still depleted, and here's the nucleus polarized. This is using EB3, so you can see where it's clustered is where the centrosome is. So without microtubules, the centrosome still collapses, the actin still depletes, and the nucleus still polarizes. So it wasn't the microtubules. So the other idea came from, again, inspired by other studies, mostly from Greg Gunderson, who's looked at wound healing where there's a nuclear rearward movement of the nucleus, which seems to be controlled by the unconventional myosin 2A. And in addition, there was an immunology lab from Max Crommel's lab who'd looked at myosin 2A deficient T cells, which they'd generated with a mouse, and found that they were unable to squeeze through endothelial cell layers. And we thought, well, maybe that's to do with the nucleus and whether you can move it or not. So we looked at myosin 2A. And what we found is that if you you can disrupt the nuclear polarization by treating the T cells with paranitroblevastatin, which is an inhibitor of myosin 2A. So you'll see that in these stills that normally the nucleus comes and it polarizes over this time scale. And if you look at the similar time scale where you've added blevastatin, you can see the nucleus remains at the rear of the cell. It doesn't seem able to polarize. Of course, just to be clear, that does not disrupt the T cell receptor signaling. We tested this in innumerable ways. This just shows two examples that we looked at the calcium flux. So with calcium flux, that occurs in untreated cells and the nucleus polarizes. If you treat with blevastatin, the nucleus doesn't polarize, but you still get the calcium flux and you still get activation of downstream phosphor work. So it's not disrupting that, it's disrupting simply the movement of the centrosome. I'm sorry, the movement of the nucleus, sorry. So the big question that kept us busy for rather a long time is, why does the nucleus polarize? Why would you move this huge, your biggest organelle in the cell, all the way to the synapse? The answer to that came as Yukako patiently looked at different pairs of combinations of markers, and she happened to look at the nucleus polarizing and the centrosome and the transcription factors. And so here is NFAT, the transcription factor, and here she's modeled the nucleus to make it very easy to see where the confines of the nucleus are. And what you'll see is that normally as the T cell comes in, the nucleus polarizes, and you'll capture the fact that it's at that point that the transcription factors seem to be translocating to the nucleus. The two seem to be connected. So she could quantitate that because you can quantitate the intensity of the NFAT GFP signal as it accumulates within the nucleus. And you can then plot this, and you'll notice that it begins at exactly the same point as the nucleus starts to move. And then you see an accumulation over time building up as the transcription factor builds up within the nucleus. So then the obvious question is, what happens if you treat with blevastatin and you inhibit the nuclear translocation? And this movie gives you the answer there. So here you can see that the T cells coming in, it's contacted the target, the nucleus is not polarizing, and the transcription factors aren't translocating. So the two really do seem to be linked. And again, Yukako could quantitate this, and she could show that compared to the DMSO control, where there is translocation and increased signal for the NFAT in the nucleus, when you treat with blevastatin, it doesn't really change at all. So this suggests that actually it's the nuclear polarization is somehow facilitating and coordinating the translocation of transcription factors that will initiate that early burst of transcription. Of course, you know, the blevastatin inhibitors are great, but actually CRISPR is in some ways more convincing because you can't have any off-target, well, you don't get the same off-target effects. And so Martin Lembach-Stokin in my lab had finally got really nice CRISPR working in the T cells. And he was able to knock out myosin 2A very effectively. And then he could show that in the control, whereas you get this translocation of the NFAT into the nucleus, you do not get that. It's severely inhibited when you knock out myosin 2A. And what was really pleasing was that when he did a killing assay with these cells using Insight, you got this depletion of the ability to kill, which sort of mirrored what we'd seen when we treated with actinomycin D. So it was all fitting together with the idea you need the myosin 2A for the nucleus to move, for the translocation of the transcription factors, and to get this early burst of transcription. So it's interesting. How does this work? The answer is we don't quite know right now, but of course in other systems, external forces have been shown to increase transcription factor nuclear translocation by flexing of the nuclear pore structures. And for that, as one of our first speaker will know, I'm really delighted to be now next door to Megan King and Patrick Lusk, who are real experts in looking at nuclear pore structures. And so we will try and do that. But it is interesting to speculate that when you look at these are T cells, they're just in a little dish. Right? There's no pressures on them, but they are somehow creating their own pressures. So you get this remarkable distortion of flexing of the nuclear membrane. And so that's also something that we're really intrigued to look further at. But we could then ask, well, what happens to early transcription products if the nuclear polarization is perturbed? And so we focused on two of these, the gamma interferon on T and F, which were products that can contribute new pathways of killing. And Claire Marr in the lab looked at this by RNA-seq, and she could show that transcriptionally, these were two of the transcripts that were inhibited. So if you treat with paranitroblevastatin, it's too difficult to do with knockout, but the blevastatin is very good for this. And so you can get a very clean answer. So you can see that these are reduced transcriptionally. And she could also then look at the protein. And for the protein, you get secretion of these products when you stimulate with CD3, which is T cell receptor. So you can see that's increased, but that is less if you treat with a paranitroblevastatin for gamma interferon and for TNF alpha. So this really suggests that nuclear polarization facilitates expression of these two products for sure, and it boosts the additional mechanisms of killing. But what does the nucleus, why does it have to contact the plasma membrane? You know, we were still puzzled by why would you go to such extreme lengths? What's special about a polarized nucleus? And so Jane Stinchcombe had done these electron microscopy tomography images where she had managed to capture the nuclear membrane, which is shown in gray here. And then this is the T cell membrane. So in magenta, it's just the membrane where there is T cell receptor clustered, which it's shown in green. And if I start this, what you'll see is as you turn this, you can see the nuclear pores. And from some of those nuclear pores, you can see extending endoplasmic reticulum in purple. And then very neatly following this were the most beautiful Golgi stacks with all in order, with the trans-Golgi network with Clathrin coat contacting the membrane at the point of T cell receptor clustering. It's quite a remarkable. I've never known electron microscopists be able to pass by a Golgi stack, and here's a beautiful example which Jane Stinchcombe lighted upon. So really what this tells us is that by bringing the nucleus all the way to the synapse, it brings the nuclear pores close to the synapse. The ER and Golgi are aligned perfectly to provide a very direct path to the synapse. So in a sense, what's happening is it focuses secretion of newly synthesized proteins at the synapse, and it gives them the shortest, most efficient route to get there. So, and I think you can see that more clearly here in these stills. So you can see the stacks here very nicely. So, and as a result, then it means that, for example, the TNF alpha and gamma interferon are captured in the Golgi. These do co-localize with the Golgi, that they're focused at the synapse very neatly. So in summary, what I've tried to show you is nuclear polarization activates an important early burst, transcriptional burst in T cells. It focuses secretion of synthesized proteins at the synapse. And what I want you to remember is that T cells, they're serial killers. They're doing this over and over again. So that pathway is going to be pumped up as T cells move from one target to the next. And for biologically, it's important for enabling infiltration, recruitment of CTLs, and boosting killing. So I'd just like to acknowledge the people who led this study. So Yukako Asano was really the person who led it, together with Claire, Adam, and Martin, and former lab members Jane and Ariane Arion Richard. And I'd be happy to take questions.
Thank you. That was beautiful.
Thank you.
And absolutely, if you see the Goldie, you can't walk away.
Exactly.
I see Hannes already has his hand up. You need to.
Yeah, great talk. So obviously the question is the nuclear repositioning just there to bring the Golgi to the membrane? And could you just bring the Golgi to the membrane easier without bringing the whole nucleus there, or is it just how things are?
That's a really great question. We've always just worked on the assumption that Golgi likes to hang around the microtubule organizing center and centrosome. Yeah. We don't know for sure that is true. We've always just assumed that. So now we need to look at that. We don't know the answer to the question. I agree. It certainly positions it. It forces a very defined structure that Jane Stinchler has noted for decades. That you have it's all a bit offset, and so partly it's offset because the nucleus is there polarizing, but the centrosome was always to one side. But, yeah.
Thanks.
Sorry.
We have two questions in the two questions that have been submitted. One is from Kevin Scrothers, who says, "Have you examined the timing of degranulation in this sequence?" So he says Salvatore Valitutti reported that degranulation can occur before centrosome polarization. So how do you evaluate the relative significance of degranulation events that occur before polarization of the centrosome compared to those that occur after the centrosome has polarized?
Yeah, we don't. I think there's a difference that was quite an old paper of Salvatore's, where he was only able to image in a single plane. So it was always very difficult to know whether the centrosome was really polarized or not. So we never see the two disconnected. So it has to be after the centrosome is docked, according to everything we've ever done. But we don't see anything different.
Okay, we have a question from Barbara Thanos, who says, "Gillian, beautiful talk. You mentioned Elosegui Artola's paper. One of the changes they see is YAP nuclear translocation. Do you see anything like this? And would the timing of the activation allow you to do this?"
Yeah, no, it's a really interesting question. So one of my new would-be PhD students who's just joined me here in Yale. I've got half a lab in Yale, half a lab in Cambridge at the moment. Wasn't the plan exactly, but so she's planning to try and look at a whole series of transcription factors to see what the timing does the timing vary? Are there some that are earlier than others? So yeah, there's lots of cool things to do, but I don't know the answer yet.
Felix.
Yeah, thanks, Gillian, for the very nice talk. So following on the mechanobiology question, so you showed that nocodazole does not inhibit the nuclear polarization, but have you tested whether it inhibits the translocation of the transcription factor? Because maybe the microtubules are necessary, to poke or to apply forces into the nucleus that lead to the translocation of NFAT. Have you tested this?
I'm going to write that down. That's a very good experiment. I don't think we've done it.
It's easy experiment, so.
Yeah, exactly. So Yukoko won't mind that one. Yeah, I don't know. I don't think we did the experiment. Yeah, good question.
Thank you.
We have another one from Kevin Scrothers, who says, "Thanks for your great presentation and response to the previous question. The next question is, how important is the volume of cytosol between the nucleus and the immunological synapse? Might a smaller volume speed up translocation?"
Oh gosh, now that's an interesting question. A smaller volume, yeah. We don't have enough data to know that. We haven't addressed that directly. I did have one last slide actually which it doesn't exactly address it. It kind of does though. Let me see, where's the share button gone again? Share, there we go. Let me try it one more time. There was one more. Here's one more. Here you go. So this is a quote from my husband who when looking at some of my images said, "Hey, the synapse is more like Times Square than the Midwest," which works now. I'm in the US. But it's a really, actually we haven't quite shown the nucleus here because this is obviously a limited section using electron microscopy. there's not a lot of space there. There's a lot packed in. So I don't know. It's a simple answer. It's really it's a cell that loves to optimize, and it's optimized delivery and killing. And it does. I mean, because it's so quick, it changes again and again. I'm really impressed with the cell.
I think it should be possible to find the machinery that links the nucleus to the plasma membrane.
Yeah.
That'd be just so exciting.
Yeah.
Maybe a split biotinylation tag or some.
We tried many years ago to tether the centrosome to the nucleus, thinking that would tell us whether it would needed the nucleus to, and actually, of course, both ended up at the synapse, and we're like, oh, that's that. But it didn't occur to us it was because the nucleus was going there anyway. You only see this by 4D imaging. It's been missed for decades. Yeah.
Okay, Daniel St. Johnson asks, what is the relationship between the myosin-dependent migration of the nucleus and the clearing of actin from the synapse?
There's none as far as I know. So the depletion of actin occurs. It very clearly occurs even when you inhibit with myosin 2A or you inhibit with knocking out the myosin 2A. So those are unconnected. That I can answer.
And we have a question from Shubham Kumar, who asks if you know if any of the Golgi proteins affect the nuclear polarization.
I don't know. I mean, there are some very obvious tethers that we're beginning to look at, and of course, again, being right next door to Patrick Luskin, the Luskin lab, as they call themselves, it's really there's a sort of a lot of experiments that we'll be able to do looking at tethers and the lamins and etc. So yeah, fortunately there's lots to do. If only we could get the whole, we don't have quite enough equipment to start experiments here yet. I think the pipette men are arriving from Cambridge today.